miRNA presents miND, microRNA next-generation sequencing data pipeline, a unique next-generation sequencing workflow for absolute quantitation of microRNAs and other small RNAs in any biological sample and species. miND is a combination of a novel small RNA sequencing workflow and bioinformatic pipeline for absolute quantitation of microRNAs in any biological matrix and species. The miND workflow uses proprietary miND spikins that are added to an RNA sample during the next-generation sequencing library preparation. miND spikins consist of 7 oligonucleotides with a unique design that reduces sequencing bias. miND spikins are provided in a specific ratio to cover the broad concentration range of endogenous small RNAs. The miND data analysis pipeline processes next-generation sequencing raw data and compiles all results in a simple but comprehensive report. The entire miND small RNA sequencing workflow is provided as a service by miRNA. Get your quote via www.miRNA.com/small RNA sequencing services. miND spikins can be purchased as a product to be included in any small RNA sequencing project for quality control and data normalization. Now let's have a look on the miND report. The data that were used to generate this report were part of a study published in Cardiovascular Research by Gutman et al. With the title Association of Cardiometabolic MicroRNAs with COVID-19 Severity and Mortality. miND report is delivered as an HTML file. On the left side, one can see a panel that introduces us to the structure of the miND report. First, it contains introduction. Second, data exploration. Third, differential expression analysis. Fourth, summary of differential expression analysis. And fifth, appendix, containing methods, R-section information, and references. Let's go through the steps one by one. Let's start with introduction. It contains important information about the project, such as project ID, customer, project manager, and when this report was generated. It also provides us with information about analysis parameters such as species, sequencing adapter, minimum read length, reads quality cutoff, significance level, and the lot of miND spikin. The second tab of the miND report contains information about data exploration. It gives a general overview of the sequencing data. 2.1 is a sample table. It contains information about customer ID of the samples, file name, and groups. This data can be downloaded in a form of a CSV file or an Excel file. They can also simply be copied. 2.2 contains information about raw data quality control. This data allows to evaluate the quality and check the data for common sequencing problems. 2.3 Reads classification. Reads classifications provides a general insights into the type and origin of all sequences that were obtained for each sample. They contain the following plots. 2.3.1 contains information about absolute reads distribution among the samples. For each sequence sample, one can see how read counts were distributed across different types of molecules, such as microRNA depicted in red, tRNA depicted in green, piRNA in yellow, etc. This plot also depicts unmapped and unclassified genomic reads. If I double-click on a specific group of molecules that are, for example, of a particular interest for me, then I would see only number of read counts that were mapped to these specific molecules, for example, microRNA, for each characterized sample. 2.3.2 contains information about relative read composition. Each graph depicts the percentage of total reads for each specific group of the molecules, such as microRNA, tRNA, piRNA, etc., within each individual sample. We can also see the percent of the microRNA, for example, that were identified for each individual sample. 2.3.3 reflects mapping statistic. The following histograms show the number on y-axis of genome mapped reads against their length on x-axis for each individual sample. The stacked bar chart visualizes the proportions of unmapped and mapped reads and can be used to evaluate the read quality. 2.4 reflects read classification table. The data in this table are equivalent to the data shown in the read classification graph above, absolute reads that we had a look before. These are raw reads counts without any normalization. This table reflects customer ID, group, and number of read counts that were mapped for each individual groups of molecules, such as microRNA, tRNA, piRNA, etc. This data can be downloaded in the form of an Excel table or CSV file, or simply copied. 2.5 microRNA mapping. 2.5.1 microRNA RPM table. This table contains all identified microRNAs in each sample. Read counts are normalized to 1 million mapped microRNAs. This table can be downloaded either as an Excel file or as a CSV file. This information can also be copied. 2.5.2 microRNA raw reads table. This table contains all identified microRNAs in each sample, and these are raw read counts without any normalization. 2.5.3 provides an overview of the identified microRNAs comparison. This graph demonstrates the amount of distinct mature microRNAs that were identified in each sample. For each individual sample, one can see two bar graphs. In red, we depict the number of distinct microRNAs that were found with the read counts above 0. In green, the number of distinct microRNAs identified with the read counts above 10. 2.5.4 microRNA read count distribution. This plot provides an overview of the abundance of a microRNA collapsed read count on the x-axis and the amount of other microRNAs in this range on the y-axis. It illustrates the distribution of microRNAs in the sample. 2.6 microRNA concentrations. 2.6.1 predicted microRNA concentrations, molecules per microliters of input RNA, and prediction intervals. This table contains information about all identified microRNAs in each sample, and microRNA reads were converted to absolute molecules per microliter based on the miND Spikin calibrator. This information can also be downloaded either as an Excel table or as a CSV file or copied. 2.7 provides information about unsupervised clustering. These heatmaps are based on RPM normalized reads and scaled using the unit variance method for visualization in heatmaps. Clustering is done using the average method of fitmap calculating the distances as correlations. 2.7.1 This heatmap shows only the top 50 microRNAs based on coefficient of variation. An additional filter was introduced here in order to increase the robustness. Only microRNAs that show an RPM in at least 1 divided by number of groups % of samples were used. For example, if we work with 4 groups, the microRNAs has to be identified with a certain RPM value above 5 in at least 25% of the samples. This approach allows to remove microRNAs that have a high coefficient of variation, but are only expressed in a very small amount of samples to bear any statistical significance or biological relevance. 2.7.2. This heat map is based on 477 microRNAs that were identified across different samples that were analyzed here. 2.8. Principal Component Analysis, or PCA. PCA uses RPM normalized microRNA reads and reduces the data dimensions down to 2, as it is presented here on the graph. 2.8.1 provides an overview of a PCA cluster by sample groups. 2.8.2 provides information about principal component analysis cluster by hierarchical clustering. 2.9. t-SNE plot. t-SNE is a nonlinear dimensionality reduction technique that is well suited for embedding high-dimensional data for visualization in a low-dimensional space. For example, here we have a 2-dimensional space. It models each dimensional object by 2 or 3-dimensional points in such a way that similar objects are modeled by nearby point and dissimilar objects are modeled by distant point with high probability. Three, differential expression analysis. Differential expression analysis uses statistical tests to find microRNAs that are over or underexpressed in a certain group. For this report, we use the well-established analysis toolkit HR. In these results are standardized. For example, we have a contrast group of group A versus group B. And then a positive log-fold change would indicate that this specific microRNA is upregulated in group A. For example, a log-fold change of 2.5 equals an increase of microRNA by the factor of 5.66. One can select a contrast below in order to view the differential expression analysis results. For example, this first group, 3.1. 3.1.1 provides information about sample overview. It illustrates which samples belong to our group 1 or group A, which samples belong to the group B, as well as explains the criteria for independent filtering. Three point 1.2 provides an overview of the differentially expressed microRNAs, and this table demonstrates only microRNAs that were significantly differentially expressed. This information can be either downloaded or copied. One can also search for a specific microRNA. For example, For example, miR15A5P. 3.1.3 provides us with information about volcano plot, and this graph visualizes the relation of the log-fold change, which reflects essentially how much did a microRNA change in the groups, and the statistical significance of this change. That means essentially that the higher microRNA would be up on this plot, they would also have a smaller FDR value, while microRNAs that were left or right from the center, they demonstrate a greater differential expression. So here in 3.1.3.1, we have an information on the volcano plot fold change versus significance. This graph can also be downloaded. And in 3.1.3.2, we provide the volcano plot reflecting fold change versus significance unadjusted. 3.1.4 MA plot. MA plot allows us to visualize the relation of the mean expression, mean of expression counts in both groups on x-axis A of a microRNA, and the differences between the two groups, log-fold change on the y-axis M. Significantly differentially expressed microRNAs with FDR below 0.05 are shown in red. This plot also can be downloaded. 3.1.5 provides us with an information about top-up and down-regulated microRNAs. 3.1.5.1 provides an overview of top 12 upregulated microRNAs between the selected groups, in our case it's group A and group B, by log-fold change. 3.1.5.2 provide information about top 12 down-regulated microRNAs between the selected groups by log-fold change. 3.1.6 provides an overview that contains the results of the differential expression analysis for all tested microRNAs. Additional TMM values were calculated by HR and are provided here at the HR test statistics table. So 3.1.6.1 reflects HR results. One can either download this information, this table, or simply search here. 3.1.6.2 provides an information on the HR test statistics. 4. Summary of differential expression analysis. This graph provides us with an overview of the number of differentially expressed microRNAs between our contrast selection, between the selected groups. For example, between normal healthy volunteers and COVID, as we can see, 41 microRNAs were downregulated and 34 were upregulated. 5. Appendix contains information about methods that were used in order to generate this report, about R session information in order to ensure reproducibility, and finally 5.3 contains references. If you have any questions regarding the miND pipeline, please contact us either through www.tamirna.com/contact or write us an email at office@tamirna.com. We are looking forward to hear from you. Thank you.